Working as a statistician at the Rothamsted Experimental Station in the 1920s, Sir Ronald Fisher was confronted with a massive challenge, interpreting decades of agricultural data to determine whether different crop treatments, fertilizers, and environmental conditions actually made a difference to yield. The data was noisy, the variables were numerous, and the natural variation in soil fertility across different plots made simple comparisons unreliable. To solve this, Fisher invented a new mathematical framework known as analysis of variance, ANOVA. The core arithmetic problem he solved was how to separate the total variation observed in a set of results into two distinct components, the variation caused by the treatment, e.g., the fertilizer, and the variation caused by random error, e.g., soil differences, weather. His arithmetic innovation involved calculating variance, the average of squared deviations from the mean, for different groups and then forming a ratio, now known as the F ratio, named in his honor. The formula is F equals variance between groups / variance within groups. If the treatment had no effect, the variance between the groups would be roughly equal to the variance within the groups, yielding an F ratio near 1. However, if the fertilizer truly worked, the variance between the treated and untreated plots would be significantly larger than the background noise within the groups, producing an F ratio greater than 1. Fisher then derived the exact probability distributions for this ratio, allowing him to calculate precisely how large the F ratio needed to be to confidently rule out random chance. This arithmetic, the partitioning of sums of squares, the calculation of degrees of freedom, and the F-test, provided a rigorous, mathematical method for extracting signal from noise, transforming raw agricultural data into objective scientific evidence.